Good morning. My name is Professor Shatineva Kunsulu from the Department of ICT. Our topic is the use of modern ICT in medicine, special medical programs. Main issues of the theme bioinformatics and biotechnology, work with databases in CBI. The purpose of our lecture is understanding the principles of organization of modern bioinformation resources and methods of working with them online and offline using personal computers. Skills of working with a different database. Training objectives, forming of ability to use the acquired knowledge to explain the results of scientific experiments. Mastering the modern means of computer processing of scientific data. Bioinformatics is an interdisciplinary field that combines molecular biology, genetic computer science, mathematics, and scientific large-scale biological problems require the analysis of large amounts of data are solved by bioinformatics from a computational point of view. Bioinformatics mainly includes the study and development of computer methods and is aimed at obtaining, analyzing, storing, organizing, and visualizing biological data. Biotechnology methods and techniques for obtaining useful products for humans using living organisms. National Center for Biotechnology Information, created by public law in 1988 as a part of National Library of Medicine and NIH to create automated systems for knowledge about molecular biology, biochemistry, and genetics, perform research into advanced methods of analyzing and interpreting molecular biology data, enable biotechnology researchers and medical care personnel to use systems and methods developed. Builders and providers of GenBank, Interest-Blast, PubMed online systems host about 1.8 million users per day at peak rates of 3,200 web hits a second. Center for Based Research and Training in Computational Biology. NCBI include PubMed, OMIM books, structured domains, taxonomy, genomes, proteins, nucleotides. The NCBI houses a series of databases relevant to biotechnology and biomedicine and is an important resource for bioinformatics tools and services. Major databases include GenBank for DNA sequences and PubMed, a bibliographic database for the biomedical literature. Other databases include the NCBI Epigenomics database. All these databases are available online through the Entries search engine. NCBI was directed by David Lippman, one of the original authors of the BLAST sequence alignment program and a widely respected figure in bioinformatics. He also led an intramural research program including group led by Stefan Elchel, David Lensman, Evgeny Kunin, Don Wilbur, Teresa Zitichka, and Zhong Lu. David Lippman stood down from his post in May 2017. In the process of database development, NCBI formed database standards such as database nomenclatures that are also used by other non-NCBI databases. One NCBI database in GenBank, the nucleic acid sequence database that contains sequence information from over 200,000 different organisms. GenBank is probably the most popular database in use and actually predates NCBI. To many, its name is synonymous with the NCBI. Here you can see the site map. Also, you can find about NCBI, GenBank sequences, submissions, support to software, molecular database sequences, structures and taxonomy, literature databases, genomic biology, tools for data mining, research at NCBI, people projects, seminars. Biocollections are curated set of metadata for cultural collections, museums, herbaria, and other natural history collections. The records display collection codes, information about the collection's home institutions, and links to relevant data of NCBI. GenBank, the NIH Genetic Sequences Databases, an annotated collection of all publicly available DNA sequences. GenBank is a part of the International Nucleotide Sequences Databases collaboration, which comprises the DNA Data Bank of Japan, European Molecular Biology Laboratory, and GenBank at NCBI. These three organizations exchange data on a daily basis. GenBank consists of several divisions, most of which can be accessed through the nucleotide database. The exceptions are the East and GSS divisions, which are accessed through the nucleotide EST, nucleotide GSS databases, respectively. Gene as searchable databases of genes focusing on genomes that have been completely sequenced and then have an active research community to contribute gene-specific data. Information includes nomenclature, chemical localization, gene products, and their attributes, associated markers, phenotypes, interaction, and links to situation sequences, variation details, maps, expression reports, homologs, proteins, domain content, and external database. Genome contains sequences and map data from the whole genomes of over 1,000 organisms. The genomes represent both completely sequenced organisms and those for which sequencing is in progress. All these main domains of life are represented, as well as many viruses, phages, viroids, plasmids, and organelles. What is genome? A genome is an organism completed as of DNA, including all of its genes. Each genome contains all of information needed to build and maintain that organism. In humans, a copy of the entire genome, more than 3 billion DNA pairs, is contained in all cells and have nucleus. PubMed, a database of citation and abstracts for biomedical literature from Medline and additional life science journals. Links are provided when full text version of the articles available via PubMed Central and other website. Here you can see how we can search, for example, protein. All results you can find here. Protein PubMed for PMID. Also you can find all articles which will include abstract. Thank you for attention. All internet resources taken from ncbi.nlm.nih.gov. Thank you for attention.